How's it going, everybody? Today we're going to be talking about gene regulation. Um, Elizabeth is going to be taking the first 3 points, and I'll be taking the last three. Um, so let's jump right into it, I guess. So control transcription initiation. Transcription initiation is the first step in the process of gene expression, where the genetic information coded in DNA is transcribed into RNA. So basically RNA synthesis, right? The control of transcription initiation is crucial for regulating gene expression and ensuring that genes are expressed in the right place at the right time and in the right amount. So there are several steps, and it begins with DNA binding. Transcription factors bind to specific DNA sequences such as promoters and enhancers. Chromatin remodeling. Chromatin remodeling complexes modify chromatin structure to allow access to transcription factors. The pre-initiation complex forms consisting of RNA polymerase, transcription factors, and other proteins. Transcription initiation, an RNA polymerase initiates transcription by unwinding DNA and synthesizing RNA. The mechanisms, we have the transcription initiation control and transcription factor binding. Transcription factors bind to specific DNA sequences to recruit RNA polymerase and initiate transcription. Chromatin modifications such as histone acetylation and DNA methylation can either activate or repress transcription. Enhancer promoter interactions. Enhancers can interact with promoters to stimulate transcription initiation. Repressor proteins can bind to specific DNA sequences to block transcription initiation. Key transcription factors. TATA binding proteins, TPB. TPB binds to the TATA box, a specific DNA sequence found in many promoters. Transcription factor 2, D. TF2D is a complex of proteins that recognizes and binds to the TATA box. RNA polymerase 2. RNA polymerase 2 is the enzyme responsible for transcribing protein coding genes. I guess we could talk about the importance, no, I mean, this is crucial. This is undoubtedly crucial. Transcription initiation control is crucial for regulating gene expression and ensuring that genes are expressed in the right place at the right time and in the right amount. If this doesn't happen, so many things can go wrong. So many things can go wrong in your body, and that would not be good at all. Transcription initiation control plays a key role in cell development and cell differentiation. It regulates the expression of specific genes. Dysregulation of transcription initiation control has been implicated in various diseases, including cancer and neurological disorders. I think the biggest is cancer. I think there's just the complexity of the mechanisms that happen in DNA and RNA. I think I mentioned this in another video. So many things can go wrong, and when these things go wrong, things tend to get pretty bad pretty quickly. So it's super important that this works. But let's jump into the next topic. Genomic imprinting. It's an epigenetic phenomenon where the expression of a gene depends on its parental origin. It involves the methylation of specific DNA sequences, leading to the silencing of one allele. So some key features: genomic imprinting depends on the parental origin of the allele. Epigenetic regulation. Genomic imprinting involves epigenetic modifications such as DNA methylation. Tissue-specific genomic imprinting can be tissue-specific with different imprinted genes expressed in different tissues. Some mechanisms here, obviously DNA methylation, histone modification, chromatin structure, just the basic ways to modify the genome. Histone acetylation in and then the chromatin structure, the modification of the chromatin structure. It all influences genomic imprinting. Some examples, IGF2 and H19. These genes are imprinted in humans with IGF2 expressed from the parental allele and H19 expressed from the paternal allele. IGF2 is expressed from the paternal allele, and H19 is expressed from the maternal allele. SNRPN gene is imprinted in humans with expression from the paternal allele. The importance here, I think it plays down to it boils down to the same things, how healthy are we? How healthy can we be? Genomic imprinting plays a crucial role in development, particularly in the formation of the placenta and fetal growth. Dysregulation of genomic imprinting has been implicated in cancer and neurological disorders. Genomic imprinting may have evolved to regulate the conflict between maternal and paternal interests. Some research opportunities here, if you guys are interested in this kind of stuff. It's obviously under-researched to identify all the imprinted genes in humans and other organisms. Elucidating the mechanisms of genomic imprinting is essential for understanding its role in development and disease. Understanding genomic imprinting may lead to the development of new therapeutic strategies for diseases associated with imprinting dysregulation. So what happens after transcription? This includes the processes of RNA processing, transport, translation, and degradation. I think degradation is super important. We'll get to that in a second. Types of post-transcriptional control, alternative splicing, the process by which a single gene can produce multiple proteins through different splicing patterns, RNA editing, which RNA molecules are modified after transcription, including changes to nucleotides or splicing patterns, microRNA regulation, small RNAs that bind to target mRNAs. It regulates their translation and degradation. Translation regulation, the regulation of protein synthesis, including initiation, elongation, and termination. mRNA degradation, the regulation of mRNA stability and degradation. It includes processes such as nonsense-mediated decay. Some mechanisms of post-transcriptional control. RNA binding proteins, proteins that bind to specific RNA sequences, regulating their processing, transport, and translation. MicroRNA-induced silencing complexes, complexes that bind to target mRNAs, regulating their translation and degradation. Translation initiation factors, proteins that regulate the initiation of protein synthesis. Sorry, I'm all over the place right now. mRNA decay pathways, pathways that regulate mRNA degradation, including processes such as nonsense-mediated decay. So what's the importance of all this? Post-transcriptional control allows for the fine-tuning of gene expression, enabling cells to rapidly respond to changing environments. Think about it, these processes happen rather quickly in your body. It's important that these things are addressed immediately. You don't want mRNAs and RNAs running around your body, creating all kinds of proteins and creating all sorts of issues, which is what leads to dysregulation. It leads to diseases. Development and cell differentiation. It plays a crucial role in development of cell differentiation, enabling cells to acquire specific functions. Again, cancer and neurological disorders. Further research is needed. It's under research to characterize post-transcriptional regulatory elements, including RNA binding proteins and microRNAs. It's all essential for understanding its role in development and diseases. Understanding post-transcriptional control may lead, again, to new therapeutic strategies. for diseases associated with dysregulation of post-transcriptional control. I mean, so you have diseases that happen in uh, pre-transcriptional control and then post-transcriptional control, and even during transcription. Um, the complexity of these processes is immense, and obviously it's under research. So genomics, I mean, genetics is the key to the future. I've said this before. Um, Gene therapy. This is all gonna. I mean, I dare say one day eradicate all diseases. Right? We can hit the delete button on a disease and have it never expressed. Um, could we even prolong human life? I don't know. The key is in genetics. But that's all I have for you guys today. I hope you guys enjoyed our presentation. It's always a pleasure to work with you guys and just share all this beautiful information about our bodies. But I'll see you guys in the next video. Later.